Good evening. The other day we discussed the cell division mitosis, where it occurs only in somatic cells and one cell becomes two. The next cell division that is very important, that is meiosis. It takes place only in the reproductive cells and one cell becomes four cells. In mitosis, one cell becomes two cells and those two cells are qualitatively, quantitatively similar with the mother cell. Whereas here, one cell becomes four cells and they are having half the number of chromosomes found in the mother cell. So now let us see the cell division meiosis. So this is a special kind of cell division in sexually reproducing plants and animals that reduces the diploid chromosomes by half, resulting the product of daughter cells. Production of haploid, this is very important. Haploid means having half the number of chromosomes. The term meiosis was coined by A.V.V.S. in 1905. Meiosis in Greek, that is lessen, becomes half, reduced, etc. ensures the production of haploid gametes in life cycle of sexually reproducing organisms, whereas fertilization results in the diploid phase. So this is nothing to worry. There you can find in a diploid organisms it happens and gives haploid, and those haploid gametes again fuse and form the diploid parent cell. That's all. Meiosis encountered during gametogenesis in plants and animals, leading to haploid reproductive gametes. During the gamete formation, this is diploid organism gives haploid gametes. After fusion of the haploid gametes, a diploid stage will reinstate. That's all. What are the essential features of meiosis? It includes meiosis involves in two sequential cycles. Whereas in mitosis you can only one cycle, whereas here you can find two types of divisions. They are meiosis 1, that is called heterotypic division, because there you can find the chromosomes and interchange of chromatin material etc. takes place. So it is called heterotypic cell division, and in this division only the chromosomes become half. And the next division, meiosis 2, this is called homotypic cell division. Nothing there, it is just like a mitosis in two cells. So meiosis 1, there you can find segregation of homologous chromosomes. Homologous chromosomes means same structure, similar structured chromosomes among which present in the cell. The meiosis 2, division of chromosomes. So in the first division, that is first meiosis, no division of chromosomes, only the segregation of the chromosomes takes place. That is, for example, if you have a 46 chromosomes, 26 like that. So here in meiosis, the just chromosomes divide, chromosomes division takes place in the meiosis 2. Now for our understanding, meiosis is just classified like this: the divisions, different phases and some phases. Meiosis divided into meiosis 1 and meiosis 2, and meiosis 1, prophase 1, metaphase 1, anaphase 1 and telophase 1. In the same way, the meiosis 2 also, prophase 2, anaphase 2, metaphase 2, anaphase 2 and telophase 2. But the very important thing here, you can find the prophase 1. Prophase 1 having a long duration, so it is again subdivided into leptotene, zygotene, pachytene, diplotene, diakinesis. You have to remember the line. For just to remember, I will give a small formula so that you can remember well. This is called LZP D square. You remember like this: leptonema, zygonema, pachynema, diplonema. In some books you can find leptotene, zygotene, pachytene and diplotene. Both are same, nothing to worry. And now let us see the different subphases in the prophase 1. So meiosis 1, it is initiated after the parental chromosomes have replicated to produce identical sister chromatids in each homologue. Pairing of homologous chromosomes, synapsis, and recombination by crossing over between the chromatids. Again, the detail I will tell you later. So these are the changes that takes place in meiosis 1. The very important thing is the synapsis. The homologous chromosomes come together. Homologous chromosomes means, for example, in human beings, out of 46 chromosomes, 23 from the paternal side and 23 from the maternal side. So in those, the size, dependent upon the size, the chromosomes are grouped together. Same size structure chromosomes grouped together, that is called synapsis. That is the pairing of the homologous chromosomes. And recombination takes place, and crossing over the genetic material between the chromosomes takes place. Everything in this first meiosis only. That is the segregation of homologous chromosomes. In the second chromosome, just it is nothing but formation of 4 haploid gametes. Each gamete differ from other gametes and also from the parent cell. That is very important thing here in meiosis. The one cell becomes 4 cells. The 4 cells are entirely different and they differ from the mother cell or parent cell. So meiosis may takes place in the life cycle at different places or during the life cycle of plants and animals at different times. Based on this, one is at the formation of spores, that is called sporic meiosis. This you can find in higher plants during the formation of pollen grains and megaspores, etc. This sporic meiosis occurs in plants having diplohaplontic life cycle. In their life cycle, haploid stage and a diploid stage of same time, same length, that is called a diplohaplontic life cycle. Now let us see that one. So here, this is a diploid organism. It produces sporangia, sporangia diploid. It undergoes meiosis. So haploid spores are formed. The haploid spores give haploid generation and produces male gamete and female gamete. Those gametes unite together, fertilization takes place, formation of the zygote. Zygote develop into diploid organism. This is a diploid organism. And here you can find this is diploid zygote. And here haploid. This is diploid. This is haploid. So haploid and diploid are equal. Those is called haplodiplontic life cycle or diplohaplontic life cycle. Both are same. So this is the sporic meiosis occurs in the spores, in the sporangium, spores. So this is called a sporic meiosis. So meiosis may takes place in the life cycle of gamete formation, that is called gametic meiosis. That is, during the formation of the gametes, the meiosis takes place. In animals, during the formation of gametes, when 2 gametes fuse and form a diploid zygote, the diploid zygote gives again this gametes, etc. So the gametic meiosis occur in plant having diplontic life cycle. Diplontic life cycle. Now let us see. So in a diplontic life cycle, the organism is diploid. Diploid organism produces a gametangia. Gametangia, there you can find undergo meiosis and form the gametes. So male and female gametes unite together, fertilization and a diploid zygote. Zygote develop into diploid thallus. So this type you can find gametic meiosis. Diploid plants and animals where you can find this one, and the haploid stage is restricted to at gametes only. The remaining you can find diploid stage. The third one is the germination of zygote. During the germination of the zygote, the meiosis takes place. That is called a zygotic meiosis. For example, in lower plants, especially in these Chlamydomonas, etc., the meiosis takes place in zygote and the resulting organisms are haploid. Zygotic meiosis occurs in the plants having haplontic life cycle. There the plant body is haploid. Plant body is haploid. As you can see here, the haploid, the thallus is haploid. It gives a gametangium, haploid. It produces gametes, haploid. After fertilization, development of the zygote, zygote only diploid. and then this zygote undergo meiosis and form haploid cells. So this diploid stage is restricted to zygote only. In the zygote only you can find the meiosis. This is zygotic meiosis, and the life cycle is called haplontic life cycle. So okay. So in the meiosis 1, you can study this one in the following phases. The first one is the prophase 1. This prophase 1 is very longer and more complex and highly significant. Further, to understand this prophase a long duration and divided into 5 subphases. Those are leptotene, zygotene, pachytene, diplotene, and diakinesis. So the first one, leptotene. Leptos means thin, that is the chromosomes become thin here, uncoil and becomes long and thinner. Each one consists of 2 chromatids. Here you can see this is somewhat late, where you can find the beginning, each one having 1 centromere and chromatid. And here you can find 2 chromatids. The next, zygotene. Zygo means 2. So homologous chromosomes come together. I told you already regarding this homologous chromosomes, similar structured chromosomes. Those are one from paternal side, another from maternal side, come together side by side, lie side by side throughout their length. This is called synapsis. So this may be also a small question to you. Synapsis means the pairing of the homologous chromosomes. So the paired chromosomes are now called bivalents. The paired chromosomes are called bivalents. So 2 chromosomes come together. In this, what happened? A synaptomal complex is formed between those 2 chromosomes. Here you can see a synapsis. So it is synaptomal complex is formed between the chromatids. The pachytene means, pachy means thick. The chromosomes become thicker and shorter. Thicker, these 2 sister chromatids of each homologous chromosomes become clearly visible. You can see clearly. Now the bivalents become tetrads with 4 chromatids. Previously you have only 2 chromatids. Now we have 4 chromatids. Each chromosome having 4 chromatids. In this region of chiasma, segments of non-sister chromatids of homologous chromosomes. Okay. So in this, remember, at some places this is a chromosome with 4 chromatids. This is another chromosome with 4 chromatids. This may be paternal, this may be maternal, and the joining of the non-sister chromatids. Look here. Here, these red, both are sisters. They came from the same chromosome. This blue color came from the same chromosome, but this red is a non-sister to the blue color. So the non-sister chromatids join at certain places. Those places are called chiasma. The point of attachment of non-sister chromatids known as chiasma. So in this chiasma, in this region, there is a chance of transfer of genetic material between non-sister chromatids. Transfer of genetic material between the non-sister chromatids takes place. Next, diplotene. Diplo means dual. Dual means each chromosome having a different segments. That is called dual. So in this, they begin to separate from each other. After the cross, here you see, after the cross of genetic material, they start separating, begin to separate from each other, except at chiasmata. Due to this separation, the dual nature of a bivalent becomes apparent, and hence the name diplotene. See, while separating, what happened? The exchange of genetic material takes place, so that one chromosome, between the non-sister chromatids, the chromatin reticulum is changed. So between these 2 daughter chromatids, here in the lower diagram you can see, there you can find the synaptomal complex is there, and here the transfer of genetic material takes place. So the red chromatid will get blue color and blue chromatid will get red color. In the same way, exchange of genetic material takes place, and in the next stage they move together, they move, separate. So in the diakinesis, what happened? They used to separate each other. The separation, look here, this chiasma, that is the point of attachment of non-sister chromatids, depend upon the length. There may be many chiasmata in each chromosome. Later they move, the chromosomes used to move from center to periphery. That is called terminalization. Another technical term you have to remember here. Immediately the chiasma move towards the chromosomes end by a process called terminalization. By this terminalization, the daughter chromatids used to separate each other. Nucleus and nuclear envelope disappear. Now the end of the prophase. The nucleus and nuclear envelope disappear, spindle formation takes place, and it is ready for the metaphase 1. So these diagrams again I am showing. This is the leptotene, where there are thin structures, and zygotene you can find 4 chromatids, and pachytene and chiasma formation, and diplotene they move to separate, and diakinesis they separate completely. The separation takes place completely. The next metaphase 1, these chromosomes are arranged on the equatorial plate. Spindle fibers came prominent. The bivalents align on the equatorial plane. Spindle fibers from opposite poles get attached to the centromere of the homologous chromosomes. Centromere to centromere you can find, and chromatid to centromere, and pole to pole you can have the spindle fibers. Then you can find 4 types. And the next one, after arranging in the anaphase, just they used to move to the poles. The 2 chromosomes of each bivalent chromatid still attached to the centromere separate from each other and move to the opposite poles of the cell. Thus, only 1 chromosome of each homolog pair reaches each pole. So they move, since they move, so look here, only 1 chromosome of the each homologous. So if there are 46 chromosomes, of the chromosome 23 only move to one pole, another 23 to another pole. So the chromosomes become half takes place in the meiosis 1, especially in the anaphase. So consequently, at each pole only half the number of chromosomes is received. These chromosomes are, however, not the same as existed at the beginning of the prophase. So they are completely different. By becoming different, the chiasmata and crossing over takes place, so that they are different from the chromosomes in the prophase. Next, in the anaphase, as I told you already, they are moving to different poles. Each chromosome consists of one of its original chromatids, and the other has a mixture of segments. Here you can see, out of these chromosomes, one chromosome is completely red, another chromatid is half red and half blue. Here that is because after crossing over. And segments of its own, and a segment of chromatid from the homologous is different due to crossing over. And telophase, it is the last stage of the meiosis 1, reorganization of the chromosomes at poles, and occur in the form of haploid nuclei, nuclear membrane and nucleolus reappear, the spindle fibers disappear, and formation of 2 cells. In telophase 1, there is no cytokinesis after meiosis. After having a little gap or immediately and enter into the meiosis 2. There is no cytokinesis. The second meiotic division may be followed immediately or after a short interphase. The DNA of the 2 haploid nuclei does not replicate. No replication takes place in this one, meiosis 1. Next, to meiosis 2, the second meiotic division is very much similar to that of mitosis in 2 cells. That's all. Because 2 cells are formed, in those 2 cells mitotic division takes place, and finally we will get 4 cells. In the prophase 2, the events of prophase 2 are similar to the mitotic prophase. nucleolus and nuclear membrane disappear. Spindle fibers are formed at each pole. The metaphase 2, chromosomes move to the center of the equatorial plane. They get attached to the spindle fibers with the centromere. In the anaphase, the sister chromatids separate from each other and are pulled to the opposite poles of the spindle due to the contraction of the spindle fibers, just similar to mitosis. And next, the telophase, the chromosomes begin to uncoil and become thin. They reorganize to nucleolus with the reappearance of nucleolus and a nuclear membrane in each pole. Cytokinesis follows and 4 haploid daughter cells are formed, and thus the meiotic division is completed. So this is the actual diagrams you can have from the cell, taken from the actual cells. There you can find these are different phases, and at the end you can find the 4 cells are formed. The next, these are the meiosis 2 diagrams, and you can have in the prophase 1, and then metaphase 2, prophase 2, metaphase 2, anaphase 2, moving and formation of 4 cells. So here you can find the difference. Here you can find one cell out of the 2, one is fully white, another is fully blue, another is completely blue, one is a small amount of blue color in the white. Here also completely white, and one has got a small amount of white. Here you can find completely white, completely blue. In the same way, the daughter cells are exactly different from the parent cells, and also among the 4 cells also we can different. That's why we are getting variations. So in a human being, those children from the same parents, they have different structures and their intelligence and have the diagrams, etc., everything because of this meiosis only. So now let us see the differences between mitosis and meiosis. So it is very important to note, it occurs in almost all organisms, whereas it occurs only sexually reproducing organisms. It takes place all somatic cells where haploid, diploid or polyploid. Whereas meiosis takes place in only diploid cells such as zygote, spore mother cells and gamete mother cells, but it never occurs in haploid cells. Remember, it never occurs in haploid cells. So next point is, it completes in one sequence and results in the formation of 2 cells, whereas meiosis completes in 2 successive nuclear divisions and results in the formation of 4 haploid cells. Next, prophase is of shorter duration, it is simple and has no subphases, whereas prophase 1 is of longer duration. It is a complex one having 5 subphases, namely leptotene, zygotene, pachytene, diplotin and diakinesis. Prophase 2 is identical to mitotic prophase. Synapsis does not take place, very important point here, between the homologous paternal and maternal chromosomes. Synapsis takes place in meiosis, synapsis takes place during zygotine stage, and the homologous chromosomes pair to form bivalents. Here in mitosis, crossing over, chiasma formation does not occur, whereas here in crossing over occur during pachytene stage and chiasma are formed. Here each chromosome consists of 2 chromatids, but here each bivalent consists of 4 chromatids. This is very important thing here. And at the metaphase, the chromosomes are arranged at the equatorial plane. Their centromeres lie on the equatorial plane and arms remain free. It's very important that you remember. Here you can find this chromosome, here the centromere, the chromosomes like this. So the centromere is exactly on the equatorial plane. But here the bivalents are arranged on the equatorial plane during the metaphase, but arms of the chromosome lie equatorial plane and the centromere are away from it. Here centromeres are away. Centromeres are away, but the chromatids are on the equatorial plate. That is the difference between this mitosis and meiosis. During anaphase, the chromosomes separate and move to the opposite poles. In anaphase 1, homologous chromosomes of each bivalent separate and move to opposite poles. And then separation That is called distinction, because they are attached and they are separating. Next, during telophase, daughter nuclei are organized. During telophase 1, 2 daughter nuclei may be organized. They are haploid. The number of chromosomes in daughter nuclei is similar as in the parent cell or parent nucleus, whereas here the daughter nuclei are haploid and contain half of the number of chromosomes of the parent nucleus. Regarding the last point, chromosomes in daughter nuclei are qualitatively and quantitatively similar. So here only you can find only one daughter. Are quantitatively and qualitatively similar to that of parent cell, whereas here the chromosomes of the daughter nuclei are qualitatively dissimilar to this because of the crossing over. Next, regarding the significance of meiosis, this is the last part of the meiosis. It helps to maintain the chromosome number constant in each plant and animal species. That is very, very important. So, for example, what is this one? If a male parent having 46 chromosomes and female parent is having 46 chromosomes, if they join, you will get only 92 chromosomes, but it will not the human being. It may be other animal or so. So, during the formation of gametes, gamete having 23 chromosomes both, and so the 46 number is maintained from each generation, that is generator's generation, so that the meiosis is very, very important. In meiosis, 4 haploid daughter cells are there. 4 haploid daughter cells are formed from a single diploid cell. This is very important in sexual reproduction during the formation of the gametes. As I told you now, this formation of the gametes to maintain the number of chromosomes. So this cell division is very, very important. Next, the occurrence of crossing over results in the recombination of genes, because the chromatin reticulum is just crossed from one chromatid to another chromatid, so there is a change of recombination of genes. So the recombination of genes results in the genetic variation. Automatically you will get a variation in the genetics. So the genetic variation is a raw material for evolution. So meiosis is very, very important. You see, there you can find recombination of genes and genetic variation, and useful in evolution. Okay, that's about meiosis. And you just read meiosis and mitosis, and read the differences between mitosis and meiosis. Thank you.